started YouTube link.
Yeah. Okay, sir.
And Pranita, you can proceed.
Yeah.
Pranita.
Yes, sir. Okay. A very good afternoon to one and all present over here. I, Dr. Pranita Gulhane, comparing this event, which is based on Indian speaking from abroad. On the occasion of 124th birth anniversary of Dr. Panjabrao alias Bhausaheb Deshmukh, founder president of Sri Shivaji Education Society Amravati, Science College Congress Nagar Nagpur, in collaboration with Microbiologist Society India, is organizing a lecture series which is based on Indians speaking from abroad. And the topic of today's lecture is career in life sciences and opportunities in South Korea. So today's guest speaker is Dr. Neha Roy from Senior Field Application Scientist, Engine Bio Company Limited, Seoul. So on behalf of the organizing committee, I would like to welcome Honorable Dr. M.P. Dhore, Sir, Principal, Science College, Congress Nagar, Nagpur. Honorable Dr. A.M. Deshmukh, Sir, President of Microbiologist Society, India. Honorable Dr. S.D. Patankar, Sir, National Coordinator of Microbiologist Society, India. Our eminent guest speaker, Dr. Neha Roy from South Korea, and all the participants who have attended this lecture series. So, firstly, I would like to introduce our eminent guest speaker of today's lecture. Dr. Neha Roy is presently working as a senior field application scientist at South Korea at Engine Bio Company Limited. There she is working on cancer diagnostics with next generation sequencing technology, as well as conducted the webinars and online trainings on identifying variants using the various panels. Apart from this, up to before this, she was working on Kangwon National University on the major research project on soybean improvement via biotechnological tools. At in his postdoctoral research up to 2020, she has worked on transcriptome analysis projects to provide insights into molecular mechanism of dwarf soybean. Apart from this, she has done a graduated research and worked as a supervisor at genetic dissection and characterization of retrotransposon in maize in 2017. She was associate research scientist at Maharashtra Hybrid Company Limited, Mahiko, up to 2014, and managed the laboratory functions including organizing, ordering of supplies and scheduling equipment maintenance. Apart from this, she has many more research paper publications to her credit in national and international journals. In soft skills, she is the communicator in delivered presentation for expert and non-expert audiences, as well as optimized and guided students with several laboratory protocols. In technical skill, she is expertise in next generation sequencing, molecular diagnostics, medical diagnostics, bioinformatics, RNA sequencing analysis, genomics, molecular breeding tools, as well as instrumentations in molecular biology. In key skills, she is the expertise in next generation sequencing, molecular breeding, molecular biology, transcriptome analysis, then RNA sequencing data interpretation. So thank you very much, Dr. Neha Roy, madam, for accepting our invitation. So now I would like to request you to deliver your precious lecture. And before that, please share your journey from India to abroad. Please, ma'am.
Thank you so much. I would like to thank Dr. Pranita for introducing me so nicely. Thank you so much for that. And I'm really honored to have this presentation and to have this opportunity to share my journey, as well as give some tips and tricks to the students who are going to view my presentation today. And I hope everybody can get something out of it. And I'm really thankful to all the our professors for giving me this opportunity. Yeah, I'll share my screen. Yeah. Yes. So to begin with, I would like to start that I started my journey from a very small town, Ballarpur. I was studying at Guru Nanak College. So I did my bachelor's in biotechnology in 2006. And after that, I went for master's in biotechnology from Nanded University. And after master's, I was able to work in molecular breeding laboratory in Mahiko. So there I started as research associate for 3 years, and then I was promoted to associate research scientist. And but still I felt that I have to do something more. So even after promotion, I discussed with my seniors what should be the next step. Of course, I knew I have to do the PhD. So I applied to South Korea, and my professor, my PhD supervisor, he contacted me and he said that your work profile and it also aligns with our lab, and then you can join our lab. My main motto was that I don't want to pay for my higher education. So the scholarship was my main aim. So for that, I had a discussion with my professor and he agreed, and I was able to get the scholarship, the university scholarship. So I'll share those details in the later slides. So with that opportunity, I came to South Korea, and since then I'm trying to survive and strive here. And yeah, from that 2014, after that, I finished my PhD in 2017. So from that, I started as postdoctoral researcher, and then again, I was promoted to research professor. But I wanted to go back to industrial background, and I wanted to move out from the academia environment. So I applied, and I got the opportunity to work as a senior field application scientist in a company. So I'll tell the details in the next slides. So what I like about our education system in India is that it covers all the basics. So when I studied biotechnology, my bachelor's and master's, I knew some part of everything: molecular biology, microbiology, cell biology, everything. So the things which I carried until now are the highlighted one: the microbiology, cell biology, immunology, and especially molecular biology, molecular genetics, and bioinformatics. So after masters, I was actually had a good long working history with this lab. This is my lab, molecular breeding lab at Mahiko. We were specialized in high throughput genotyping, and we were engaged in agri-genomics. So what is agri-genomics? So when we think about breeding and to make a new crop, so if we have a conventional method, so it's like by viewing, we see that we have to wait every generation to see visibly which is the highest producing crop, and then we will continue breeding those two crop. But this takes very long time, and we need to wait generation after generation for that. So, but when we have a molecular marker, we know, so we can directly pick those plants, and then we can breed them, and just this shortens the process. So this is the thing which molecular markers does. So from 1900 there was a big from the Mendel laws. It actually changed the path of molecular markers, and since the advent of like PCR technique and then Sanger sequencing, the field of molecular markers has completely dramatically improved throughout even today. So these days, from like the first plants that was completely sequenced was Arabidopsis in 2000, and the rice was sequenced in 2002. And from then, there is no looking back. There have been several plants that have been sequenced numerous times, and now we are doing precision breeding and predictive breeding thanks to those technologies. So in the molecular markers, we have various markers and the first kinds are RFLP and AFLP, but they are having their own drawbacks, that is they are low throughput and the tedious and very long protocols. But The newer markers are SSR and SNPs. They are abundant and highly polymorphic. And for SSR, there are many high density map have been made thanks to the new next generation sequencing technology. And although SSR are a bit laborious, but SNPs, they are like the today's markers that that everybody is focused on. So SNPs are majorly worked on these days. This is the setup of my laboratory in Mexico. When I was working there, we used to do very high throughput work. We used to do PCRs on the 96 plate, 384 well plate altogether. We had this liquid handler TCAN system. This is a robotic system, and multiple plates were set up in a single run. And in one day, we were able to do more than 2,000 samples, and we would able to do the isolation, PCR, and the final output within a day of more than almost 2,000 samples. And yeah, this is TCAN system. And then the output we use for that, I saw the journey from the gel-based system to the capillary-based system, which is here, it's Shimadzu multi-NA, and the fluid ion system. Although this system is very old now, but I was there to see this transition when I was working. Major projects that I did while working were assessing hybrid purity, evaluating rice germplasm to screen the bacterial leaf blight, and then producing genotyping results to estimate the recurrent parent genome for backcross program in rice. So after that, I came to South Korea. After almost 5 years in Mexico, I came here for PhD. So 4 years, I worked as a PhD student where my major crop was maize, and I worked on transposon elements. And after that, I started working as a research professor. In that, my major crop was soybean. So I'll just tell you a little bit about soybean. I was working in characterizing the dwarf phenotype, as dwarf phenotype is more in demand by the breeders. And also there was a project for lowering the saponin content, and then herbicide resistant and cyst nematode resistant. These were also added project that I did. And I was working on the transcriptome analysis and characterizing the genes that were responsible for these traits. And in addition to that, I worked in non-model crops. These crops, we were using the transcriptome technology because they did not have any genome sequence. And I was primarily responsible to find the do the functional annotation and to characterize the key genes that were involved in the specific components of the particular plant. So this was the workflow, a typical workflow, like from sample collection from the field, and we did the wet lab work of DNA, RNA isolation, and then we did the NGS analysis, the transcriptome and bioinformatic analysis. And after that, these are the bioinformatic pipelines. If I will not go in very detail, this is too descriptive. So after that, the transcript characterization, manuscript writing, and the experimental validation part, I used to take care of that particular part as well. I have several manuscripts published in these journals, in all SCI journals. And after that, I wanted to get back into industry. So based on the experience of next generation sequencing, I came to this company. This is Engine Bio. They are specialized in making cancer IVD and next generation sequencing kits. So, and they also provide bioinformatic platform for analyzing the huge data. And most of the kits in the company, which detect hematological malignancy, solid cancer, and oncoacute for solid tumor, these kits are mostly CE-IVD marked. This is my team, as you can see here. So A typical next generation sequencing workflow includes these steps: the sample preparation, like getting DNA isolated from blood sample or FFPE samples. Then for NGS, there is a step called library preparation. And after doing the sequencing in high throughput, either Illumina or PacBio, there is a bioinformatic analysis step and the report generation step. So my company specializes in these two steps: the making of NGS panel for several diseases, like for majorly for cancer, and we also have a kit for MTB, flu, and COVID. And then we provide the bioinformatic solutions to view the results and generate reports. These are the biomarkers that we are targeting to identify the cancer and identify cause of cancer. So there are several biomarkers that are used, like as a single nucleotide variation, gene expressions, and CNV, that is copy number variation, either copy gain or copy loss, then indels, fusion, and structural variation. So my company specializes in these four: SNV, indels, fusions, and structural variations. So how is this useful? So if we give one single kind of drug to all the patients, it might have three effects. Either they will improve, either it will have no effect, or they will go even worse. But if we have the precision diagnostic system, if we have the an analysis of the gene, whether they have it or not, and based on that, we can give personalized treatment. So this is very useful in chemotherapy. So the patients are given the chemotherapy treatment based on the gene panels which they have, like the markers which they have. So with this, everybody can have the improved or positive effect. So this is how NGS system can be applied for cancer diagnostics. We have two kinds of panels. One is amplicon based and one is hybrid capture. So the major difference between these two is that the hybrid capture generally uses those genes which have very huge size, because the sequencing, if the size is very long, it is not possible to cover based only on amplicon based assays. So I think we don't need to go much detail in this, but I can say the difference main here is the amplicon based, we have BRCA AcuTest Plus, HLA AcuTest, and MTB. And for hybrid capture, we have heme and solid AcuTest panel and OncoAcu panel. So the difference here is the BRCA is for germline cancer and breast and ovarian cancer. And HLA AcuTest is for organ typing. So when there is an organ donation, there has to be an organ typing. So where all the markers are matched and seen whether it is acceptable or not for the donor and recipient. So that can be checked using this kit. And this is the very recent kit that we have made for tuberculosis. This includes major resistant genes, and we can identify whether the patient has all the resistant genes, and then the prognosis can be given based on that. And this is for solid heme cancer and solid tumor and oncopanel. The only difference is here, the heme consists of all the blood cancer, and solid consists of all the organ level cancer, so liver and stomach, pancreatic, like that. And OncoAcu panel has a large size of genes which we focus on. So that was about the analysis, NGS before the Illumina. But how will we get the analysis results? So for that, after having the sequence run in Illumina system, either by MySeq system or NextSeq system, the FASTQ files are generated, and those FASTQ files are sent into servers. So this server is in our company, And our bioinformatic team has pipeline ready. So we analyze it here, like pipelines are also set up for each panel. So they analyze and they send out the results, and then the user can see the results in the platform which we already provide. So he can view the result, he or she can view the results and generate report using that software system. So that is the bioinformatics solution the company provides. So that was about my journey. So, but now I'll begin and I'll explain a little bit about the alternative careers or the careers in life sciences. So in life sciences, there are so many opportunities that we or not many are aware of. So if we only see in the academics, there is that technician, lab manager, and then postdoc, instructor, professor, assistant professor, adjunct professor, like that. But even after that, there are many other lines that anybody can choose. It can be from business, from communication, from finance, and every corner of the world, and every corner of the world, and have so many opportunities. I have highlighted a few here. The very first is medical writing. So while doing the master's and PhD, already we have developed our writing skills. So medical writing includes writing handbooks, protocols, and application notes, manuals. So a life science graduate can excel as a medical writer or a technical copywriting. Also, there is another field called scientific journalism. It is also similar to medical writing, but it includes other scientific fields as well, other than medical background. Then there are these two fields are related: healthcare informatics and data science and big data analytics. So these are related to bioinformatics. So if a person has a very interest in bioinformatics, computers, programming, so they can have their career in this field. Then there is a technical sales, FAS, and business development. So right now I am in a technical sales or FAS team. So basically what here we do is we provide the training for the clients. So for example, for me, the clients are the doctors and the technicians of hospitals. So I give them complete training about how our system has run and how to prepare the library and how to analyze the samples, like that. So FAS can be for any field, for plant, for medical, for other fields as well, microbiology, and everywhere. Then there is product marketing and management. This is also a very exciting field for life science graduates. We don't need to go for MBA, but a life science graduate also can go in this field. Then there is intellectual property and patent law and regulatory affairs. So these are also a very good field that life science graduates can think of. And lastly, entrepreneurship. This is also a very exciting field. We already have an example in India, Majumdar Shaw. She is doing tremendous work. She inspired so many women in India to, not only women, everybody to choose this field after life science graduation. So how about coming to Korea? So if a student want to come to Korea, Korea has so many scholarships and programs with which they can come. So I have put the scholarship links, like I have noted here like almost 5 to 10 scholarship links here. So if you see this later, you can use this. So this first one, this is GKS and KGSP. This is the most and like the best scholarship which is available here. Only thing is that you have to learn Korean for 1 year. And if you qualify a certain level, like until level 3, in 1 year, then you are awarded with the scholarship. So this scholarship includes full tuition fee and also prize money if you are really great in Korean, like if you qualify more than level 3. And then it also includes, other than tuition fee, it will give you research support, dissertation printing cost, completion of grant, prize money, and also the to and fro ticket to home country. So for that. And it is available for bachelor's, master's, and doctoral degree in all the institutions in South Korea. So every year, many students, they use the scholarship, they avail the scholarships. Approximately 220 undergraduate and more than 1,000 graduate students use the scholarship. So, and the thing which we need to keep in mind that GPA should be at least 80% in the previous institution before applying this course. Similarly, the next, this Seoul National University, it's like the Oxford University of Korea. It's the best university, one of the best university. So they also have their own scholarship, and they provide the full tuition fee. And apart from that, they provide the living expenses and of course medical insurance. This is very necessary when you live here. So they also provide that with a round trip fare. And here also they should there is not necessary to learn Korean, but if you want to learn, they can help you with the cost of the language program. And but most of the scholarship have this requirement that you have to maintain a certain GPA record while studying. So likewise, there are all the scholarship from major university that you can look for. And when you are searching for any university in South Korea, almost every national university and other autonomous university, they have their own scholarship systems. Even if it is not the university scholarship, it will be like from department-wise scholarship. So they have so many programs that see, if you see, I have linked here. This is 30 scholarship, like from 30 different university, and every university have their own program. So many opportunities are here, and these are only for foreigners. There are also so many opportunities for Korean students as well, but these are the links that I am sharing. These are for foreigners, for international students. So I would really recommend to go through this and check suitable to you and which is very close to your interest, and then you can try for this. After studying, if you want to search job here, these are the top website where with the help of which we can find job Korea, Saramin, People and Job, LinkedIn, and Brick Job. So Saramin and Brick Job, these are the Korean websites. Also the Job Korea, this is also Korean website. And of course, LinkedIn, and these are English websites you can see. And if you are already living in Korea, then you can go to the job fair. These two are especially for the international students. So next, I would like to give a few advice on the CV and resume. So I would like to recommend all the students to keep it short. The very long CV, nobody reads these days. They will just give a quick glance and they will see for the keywords. If their job requirement matches, then only they will entertain to look the whole CV. And the first page should include these information: address, date of birth, cell phone number, email, and also the previous job titles, or whatever project you did, the project titles, and the major project techniques that you do, those titles should be emphasized there. And for academic jobs, there has to be a cover letter, which is your self-introduction and a kind of your introduction of how you manage to study and why are you so interested, and what is your path, like that. A brief summarized introduction should be there in the cover letter. And research proposal. In the research proposal, there are it has different parts: introduction, problems, objective, methodology, references. So they should be proper order, and they should follow a story. And then and also I would really recommend to include Gantt chart. So it is like a small graph that you show like this many time you will require to do a certain task, to gather information, to do an experiment, and like that. So this is very useful. And if it is only descriptive, it becomes boring. But if there are tables, figures, and chart, then it appears interesting and people will read it. And last, I would like to say about the pros and cons of living in South Korea. So, in terms of safety, I would say it's a really safe country, especially for women who are in India. They want to come here. Like I remember working in my lab until late nights, midnights, but I used to go home without worrying about anything because it is a very safe country. And like technically it's still in a state of war, but it doesn't matter here. Like the people who are here, they don't seem to be affected by that. It's only the news channels and the media that are exaggerating that information outside. Next point is accommodation. The students' rooms and all, they are super furnished and they look very good, futuristic kind, but they are very small, cramped, and expensive. So when you apply here, you have to keep that in mind while coming. Like India has bigger houses and open houses. In South Korea, they are very closed houses, so you will get a kind of culture shock when you come here. Cuisine, food wise, there are abundance of traditional fare, but there is a lack of comfort food. The food wise, many people are suffering here, especially vegetarians. They find very hard time when they come to South Korea. Culture wise, it's a very closed society, but they are also very helpful. So it really depends on how you find people, how they are around your environment. But yeah, the expat communities, the foreigners which are here, they are very helpful. They look out for each other. So we all have several groups. So we are very helpful and accommodating. So everybody reach out to everyone. Then work culture is a very palli culture. Palli is a word for in Korean, which means to do everything very fast. So, but yeah, work culture wise, they are friendly and amicable, but sometimes the scheduling becomes very unpredictable and work environment can be high pressure work environment. But I believe the high pressure work environment is everywhere over the world. So, but yeah, in Korea, it can be difficult sometimes. Cost of living, the basic amenities are really very convenient. Although the gadgets and clothes, they are expensive. It is not same like US or anything. Education and school wise, yeah, the schools are excellent here. But yeah, the stressful environment is there. Like we have to be ready for unpredictable workload or anything. And healthcare system is a very plus point here. Like they are very cheap healthcare and everybody's taken care. And these, like when I came, the insurance that I had, it did not cover many things. But when like 3 years before they started, like every student has to take the national insurance. So it includes almost everything. So healthcare wise, Korea is having very nice way to take care of everybody. And yeah, that is very good. But if you're living in a very remote area, so coming to a location, main city can be a difficult thing. But I think mostly the universities are located in a very city places, and university have their own hospitals as well. So I think healthcare wise, this country is a very good country. You can think of it. So out of this, the main point is when you come to South Korea, you will feel a culture shock. And it's not that everybody speaks English. There will be, when you come here, you have to learn Korean a little bit to manage. So if you learn Korean, you will have a very comparatively easier life. But yeah, it is difficult. But when you come here, when you get into your studies and when you go through the life becomes very easier, as it's not that difficult. Like I would say, I was really thinking to go to US, but I don't want to go to US anymore. I want to stay here now. So yeah, that was my concluding slide. Thank you so much. And this is so lastly, I would see like success looks only small, but we have to do so many hard work for that. We have to be persistent, face so many failures and disappointment, but we have to get back, bring back discipline, do the hard work. We have to keep the dedication on, and then that's what brings us this success, which what people see. This is not visible, but this is visible. So that is how I would like to end my talk. And for any inquiries, you can go to my LinkedIn and you can contact to anyone from our society and they can relay to me and I will be really happy to help anyone. So thank you so much. That's all from my side. Yeah. If there are
Thank
any
you, ma'am.
questions, I would be happy to answer.
Yes, thank you very much, ma'am, for your precious lecture based on the topic of career in life sciences and opportunities in South Korea. You have really given a very detailed idea about the fellowships and the websites also, whatever the students need to pursue their higher qualifications there at South Korea, as well as you have given other ideas also, the food quality, the schools and everything. If anyone want to go there and settle there in South Korea, and really you have emphasized your knowledge and shared your knowledge with all of us, it would be very great for us. So thank you very much, madam, for your guidance, for your information. Now I think if any participant would like to ask any query, they can please put their query in the chat box so that we can ask ma'am. And ma'am, it is a request to you that if you could write your email ID in the chat box so that if the participants wanted to contact you regarding any query, they can mail you.
All right. Yes, I'll write it down.
Yes, ma'am.
At the outset, I want to appreciate Neha really giving a very, very good information. And
Actually, I
school,
knew like what a student would think and what would he or she would want to have. So I keep that in mind and I made those slides.
correct. So really you have given a very, very good information, and with your information, if a single student got a success to enter in Korea, I will consider it is a success of today's lecture series. So really, I appreciate you, Neha. And you give your Indian address. We'll send a memento on your Indian address.
I will
Okay?
take that.
Thank
I will take that memento at Ballarpur because
Very good.
I visit Ballarpur in the Ballarpur Institute of Technology in the food technology department in every month. So still I am.
Oh, really?
Yeah, I am consultant there
Wow.
and also adjunct
Okay.
professor. Yeah.
Wow. Okay. I will be happy to take it from you.
Neha, I want a
Yes.
couple. I want to ask couple of questions. One is regarding your kit, that is regarding the
Yes.
Mycobacterium tuberculosis.
Yes.
Yeah. See, Mycobacterium tuberculosis, it is a big problem in India,
Yes.
and this particular kit, how it will help in India? Are you introducing that kit in India? How far it is possible?
It's a very new product and we are now beginning to start the marketing of the kit, MTB kit in India. So the plan is like it is an NGS kit. So we have to market in those places where the NGS facility is available.
Okay.
So in TB the problem is many people don't go for NGS analysis because it's very expensive. But the major hospitals which have a large number of patients. So in those hospitals, I think there is a big market for an MTB kit. And I recently did the study of all the NGS kits that are currently available. So I think the kit that our company has formalized and made, it has the maximum number of genes and maximum number of uh, resistant genes markers. So I think it will be really success if we are able to
Yeah.
make some space in Indian market.
Because mostly in India, we are speaking even in the course, in the MSc biotechnology course or in the biomarkers are taught, and for the particularly in the diagnostic medical biotechnology, where they all the things they are based only on what on the biomarkers, particularly the interferon gamma release assays, and people are talking on those lines still in India.
Yes.
Yeah, so
So
you can
actually,
understand what I want to say, because they are not
yes.
very competent one to detect early detection of tuberculosis or in the asymptomatic or the such type of the tuberculosis, because many more people they are suffering from actually the extrapulmonary tuberculosis from the good families also. It is not the question of the poor or the such type of the people, but I know the cases. I know the cases who suffered from the tuberculosis, which are from the affluent families, and they could not get detected. This is the problem. And one more thing regarding the cancer kits. The cancer, you are going for the treatment. Your cancer kits are determining the treatment direction.
Yes, prognosis. It's
Yeah,
it's in
yeah,
prognosis.
yeah, prognosis. But early detection, any early detection?
Yes, the BRCA kit, that is the
BRCA,
early detection kit.
yeah.
So in that, the biomarkers that are detected, so it will identify if the patient has the biomarkers or not.
Yeah.
Like for example, Angelina Jolie, she her grandmother and her mother all were suffered from breast cancer. So she did that
Yeah,
Illumina kit.
yeah,
Yeah, so she understood she has those biomarkers, so she is
yeah,
like she can get the breast cancer
yeah.
anytime. So
Breast
she
cancer,
did.
breast
Yes,
cancer is a very big problem nowadays in India. And
yes.
why it is it because of environmental and the other type of the thing, or the epigenetic problems are there, or they are the hereditary problems are there, so that we cannot, because if you, in the Ballarpur, particularly in the Ballarpur, you know how is the pollution and all other type of the things. And many more
Yes,
cases I
my
saw.
mother was the victim, Professor.
Yeah,
So,
I know actually
yes,
all these things. You know, there
yes.
was one Ved Prakash. He was working in our sweeper was there. His wife was also used to come.
Yes.
She died because of breast cancer, and there are so many
Oh,
cases. So many cases are there. So is it
it's
a
really
is it epigenetic,
disheartening.
or is it epigenetic, or is it genetic one? So how you can differentiate this particular thing? Any such type of the markers are available, or is your
Yes.
company is working on that?
So my company basically that produces their gene analysis. So
Yeah,
they identify the genes. So if they are genetic, so like in a patient, the reasons can be numerous. It can either be a genetic background or environmental stress. So with our kids, we can find out what are the reasons. How like is it a genetic? Is it a germline mutation, or is it just a
yeah,
somatic mutation
yeah.
like that?
Germline mutation. Yeah, exactly.
Yes.
So
So if it is germline mutation, then it is from familial, it has come
yeah,
from the familial
family.
background.
Right, right.
Yes. So it's like we
So
can
they
find out like who are the more prone
prone
cases,
people.
like can get the diseases. That
You
is
can
what
have
we are able
you
to.
can have the SNPs there. You can use for that.
Yes, yes.
Yeah.
So we identify majorly the variants, the
Mhm.
CNVs, the copy loss and the copy gain. So that we identify. And so basically what happens in cancer is the cells starts growing tremendously. So what our kids are doing here, like they identify the gene if it is working or not. So if it is defected, so that is causing the cancer. So that early diagnosis is possible with
Okay.
the kids. Yes.
And one last question it is there regarding the entry in the South Korea. What is your opinion that before coming to the Korea as such, is there any necessity or is there any requirement that the student should have the experience in the company or in the industry? How it
Korea is a very academic prone country, and actually Korea has a very high regard for Indians. So
Yes.
that I actually missed to say in my presentation. So in South Korea, Indians are really they are chosen. Like if we find an international student we want from India, it is like that, because we have a very good theoretical knowledge. So academics is very important while coming to Korea. So especially for the young students, academics is very important. Industrial experience, it really helps because it gives you the exact knowledge of what are the current technologies going on. But having a sound background, having a sound basics, the clear basics is very necessary when we come to South Korea.
And in
because
that
after
India,
coming to South Korea, in universities, the education, it is not focused much on educating the basics. They are focused on doing the projects.
Applied
Yeah, and then they really don't give much emphasis on doing the writing work or like studying that. They don't think that they will give you extra time for that. You have to do it on your own. So before coming to South Korea, I would advise all the students that they should have the basics clear.
And in India, we are giving much more on the basics.
Yes.
So I will interact regarding your NGS and your kits because we are interested in that.
So if in that case, like we can have a discussion briefly, specializing in one panel, and then we can give all the details if with whatever is needed.
Because there is one institute at Nagpur, you might have heard, Central India Institute of Medical Sciences.
Yes.
Yeah, where Dr. Daginawala.
I did my some
your project
master's project I did there. Thanks to you. Yeah, thanks to you.
I think you went for on-job training there.
On-job training at that same institute. Yes, I went to.
So now they have developed so many kits, and they are in the medical diagnostics. And Dr. Daginawala is still there at the age of 90. He is still working. Yeah.
So inspiring.
Yeah.
Wow. I met him when I was there.
Yeah. He is still there, but his age is now 90. And he came to Ballarpur also to our college in the surveys of chikungunya. So yeah.
So it's very inspiring to hear that.
So really. Actually, they are very much interested in all such, particularly in tuberculosis and other extrapulmonary tuberculosis. And I am in good touch with them. I am in association.
We can have a detailed discussion just on tuberculosis.
I will talk with the research director, Dr. Rajpal Singh Kashyap.
Yes. Dr. Rajpal. He's also still there.
He is now director. You remember?
Okay. Yes, I remember him. Okay. Yes. So nice to hear all those names. Actually, I feel like my age is reduced.
Yes. Very good, Neha. Very good now.
So
now,
thank you once again.
I request Pranita.
Yes, sir.
If there is no question from any other member, you can conclude it.
Yes, there is one question in the chat box, ma'am. That participant is asking that when you first visited South Korea, what problem did you face while studying?
Language.
Okay.
So thankfully and very fortunately, my professor spoke Korean. But the university where I went, majority of professors, they were only talking in Korean, but they were kind enough to help me by, like, I can give my exams and my tests that I can do in English. But the classes that were going on, they were in Korean. So I only had like few lectures in English and rest everything was in Korean. But yeah, you can give the exams in English, so that is fine. And the second problem was the weather. So I am from Ballarpur, the hottest place in India. And I came here and there is snow here. So, and it's 8 months there, it's cold, and just 2 months summer. So weather was also very tough for me. And I was also, I was a non-vegetarian, so the eating wise, that was not a problem. But the people who are vegetarian, they face very difficulty here. So like even the definition of vegetarian is very different here. So the pickles, they have that they are vegetarian, but the pickles, the ingredients are not vegetarian, but they don't understand that.
Okay,
They
okay.
will say to Indians, it's vegetarian, you can eat it.
Yes, yes, yes.
So yeah, that was another problem I faced. Yeah.
Okay. So thank you very much, ma'am, for your detailed information, what you have given in today's lecture. Really, you have emphasized a lot on the career opportunities in South Korea, as well as the problems which may be faced by the people who may visit to South Korea. And your experience
So
also you have
I
shared.
would like to, I'm sorry, I would like to just add one point here to the students. So when you come to South Korea to study for higher studies, there will be times where you will feel very lonely, because it gets like that. The whole winter is very lonely and depressing period. But you have to keep in mind and keep moving, and finally you will get to your final goal. So coming to South Korea, you can be lonely because you're away from your family, and the time in India and here is different. So you are not able to communicate with your Indian friends and family all the time. So you can get really lonely here, but you just have to remember and keep moving. And yeah, the internet is very good, so you are always online. So that is like a support system here. And then yeah, you will finally reach to your goal. So yeah, that is my point I would like to add for all the students.
Yes, that
Thank you
is
so
very
much.
much
That's all.
important for the students, because
Yeah.
that is one more thing, that if they are going abroad for their studies, then this must be work out. So it is very inspirational, ma'am, that you have shared your experience too with our
Yeah,
participants,
thank
with
you.
our students. It will be inspirational to them. So thank you very much, ma'am, on behalf of the organizing committees, Honorable Principal Professor Mahendra Dhore Sir, Science College, Congress Nagar, Nagpur, and Honorable President Dr. A.M. Deshmukh Sir, President of Microbiology Society India. On behalf of the organizing committee, we are very much thankful to you for delivering your precious lecture. Thank you, ma'am.
Thank you.
Thank you. My pleasure. It's my honor. Thank you.
Thank-
Bye-bye.
Bye-bye.